My name is Matt Lozancich, and I will be doing my presentation on chemoreception, smell, and taste. Today's date is 6/22/22, and there's my name. All right, let's get on into it. So, chemoreception is a physiological process whereby organisms respond to chemical stimuli. In humans, there are two major chemosenses, smell and taste. Smell is called olfaction, and taste is called gustation. Olfaction. Olfaction is the sensation of smell that results from the detection of odorized substances aerosolized in the environment. And then olfaction allows us essentially to discriminate between millions of different odors. So if you had a pungent flower, you would have particles that are being released or coming off the flower, and they would be aerosolized by the environment, and then they would be uptaked into the nasal cavity, and certain receptors would recognize these and trigger action potentials. And then based on the response that we get from that, we can determine what type of smell it is or what recognize the type of flower. So olfactory anatomy. The whole nasal cavity is lined with olfactory epithelial cells, and an olfactory epithelial contains olfactory receptor cells. From these receptor cells, cilia emerge, and cilia spread over the surface of the olfactory epithelium. Basically, what the cilia does is it increases the surface area to trap odorants as they enter your nasal cavities. And this is why you can recognize so many different combinations of smells. Odorants enter the nasal cavities. They stimulate the receptors in the cilia, depolarizing olfactory receptor cells and initiating action potentials, which travel up through the axon and into the olfactory bulb, right up here. The axons that travel to the bulb have to go through the cribriform plate of the ethmoid bone, so this bone right here, so they have to penetrate through there. And the axons that travel from the epithelium to uh, bulb is called the first cranial nerve. So now we have the olfactory bulb. So, like I said, you have the receptor cells that penetrate through the cribriform plate and the ethmoid bone, and then olfactory cells converge on the structures called glomeruli, which are these things right here. Boom, boom, boom. In glomeruli, the receptor cells synapt with matrial cells and tuft relay neurons. Both cells project into the olfactory tract, which is right here, which is basically just a bundle of fibers that carries olfactory info to the olfactory cortex down this way, and most olfactory processing will occur in the olfactory cortex. So we have the olfactory cortex. The olfactory cortex is a collection of cortical areas that receive information from the bulb. The olfactory cortex consists of the prefrontal cortex, the posterior orbital frontal cortex, the amygdala, olfactory tubercle, and the perihippocampi gyrus. The piriform cortex is the primary olfactory cortex. Is the primary olfactory cortex that is involved in the conscious perception of smell. This includes the anterior olfactory nucleus, the piriform cortex, regions of the amygdala, and then the preamygdala complex, and the rostral and terminal cortex. So now we're on to gustation. Gustation is the sensory detection of food on the tongue. This is separate from olfactory, which was about smell, the processing of smell. Now we're on to the processing of taste. So the sensation of taste occurs in the mouth when a substance reacts with a taste receptor cell located on the taste buds. So now the kind of like anatomy and the physiology of taste buds is that taste buds contain four um, types of cells. You have type 1, type 2, type 3, and the basal type 4 cells. Type 1 are kind of like glial-like support cells. Type 2 are receptor cells. Type 3 are prenaptic cells. And then type 4 are basal cells, which are precursors of taste receptor cells. Each taste receptor cell is non-neural polarized epithelial cells tucked into the epithelium with a tiny tip protruding from the oral cavity. Um, and then to kind of dive in a little bit more, taste is comprised of five basic sensations. You have sweet, sour, bitter, um, uh, salty, and umami, which umami is just like savory. So sweet and bitter and umami taste are associated with type 2 receptor cells. And these cells in particular contain a lot of G protein coupled receptors. Sour is associated to type 3 presynaptic cells. Um, the protons from the acidic food activates ion channels. So this is less G protein and more ion channels in the presynaptic cells. And then salt is also believed, they're not entirely sure, to be associated with type 1 support cells, but this also activates channels. And then the receptor cells for taste are located mainly in the taste buds. Receptors are called taste receptor cells. The taste substances must first, to taste a substance, it must first be dissolved in the saliva and mucus of the mouth. The tastings then interact with the protein receptors on the taste receptor cells. Interactions called signal transduction cascades. Unless this is G protein, then it typically goes through that process. Casual chemical messengers, well, casual chemical messenger molecules will be released from the taste receptor cells. These signals activate primary sensory neurons, whose axons run through cranial nerves 7, um, 9, and 10 to the medulla. The sensory information passes through the thalamus to the gustatory cortex and the insula. And sensory information compares the inputs and interprets the sensations. And then based on that, these signals will elicit responsible or elicit a emotional response. So like, oh, that would tasted good, or ooh, that would tasted bad. And that's it. That is it. And then I will be posting an assignment for you to go through too.